Today, I would like to talk to you about some advances we've been making in my laboratory toward the analysis of progressively larger RNA analytes. And why are we so interested in RNA? RNA can be a very appealing therapeutic target. As you might know, the human genome contains 3 billion bases, and out of these 3 billion, only 2% of these sequences code for proteins. Nevertheless, these proteins account for over 10,000 drugs that have been approved over the years. At the same time, the remaining 98% of the genome information might or might not be transcribed into non-coding RNAs, and these RNAs account for fewer than 10 drugs that have been approved thus far. Therefore, we are looking at the very extensive druggable space that needs to be explored. In addition to that, RNA can be considered as a therapeutic agent in its own right, as demonstrated by mRNA vaccines of recent fame, as well as up-and-coming strategies based on RNA aptamers, silencing RNA that can be used to shut down the production of different proteins, or replacement protein strategies where the mRNA provides the code for producing proteins that are not made by a certain cell due to some malfunctions. Therefore, there is a huge interest in exploring RNA, and mass spec can play a very important role in this RNA drug discovery. In particular, mass spectrometry can be used for the identification and the structural characterization of natural RNAs, as well as synthetic nucleic acid produced in a lab. There are many examples where mass spectrometry has been used to perform the functional elucidation of these RNAs, or also to screen for ligands as possible inhibitor candidates. And as we go toward the production of nucleic acid therapeutics, mass spectrometry will sure find increasing applications in production support operations like quality control and quality assurance. A typical analysis that we can perform by mass spectrometry is a molecular mass determination. I'm giving you here the example of an electrospray mass spectrum of an oligonucleotide of about 124 nucleotides. This construct was produced in vitro to mimic the packaging signal of the HIV-1 genome. And you notice we have a nice charge state distribution right here. The sample was submitted to extensive desalting, so we don't see too much desalting in there. This analysis was performed by nano electrospray on a Fourier transform ion cyclotron resonance instrument. So we can take advantage of the resolution afforded by this analyzer to resolve the contributions of the different isotopes. Based on the spacing between contiguous isotopes, we can infer the charge state. In here, in this case, this is a minus 40 charge state. Based on this information, we can come up with an experimental mass that is greater than 40,000 mass units. This mass is very close to the predicted mass, which we can calculate by using the sequence. And in this case, the discrepancy of only 5,000 units amounts to 123 part per billion accuracy. So with this type of accuracy in molecular mass determination, it will be straightforward for us to distinguish a single point mutation, let's say a cytidine being replaced by a uridine, that will cause a mass shift of 1 Dalton, which could definitely detect just by performing a mass determination like this. The next step for the characterization of this type of product will be to perform a tandem mass spectrometry experiment to get confirmation of the sequence. In this case, we isolated several the precursor ions, several charge states produced by the packaging signal to obtain a better ion statistics. And we submitted these to collision-induced dissociation in the collision cell of our instrument. As you notice from the product ion spectrum here, there are still the remnants of these precursor ions. We decided not to obliterate them completely because we were still able to detect sufficient signals here to provide the information that we were after. So if we zoom in different regions of the spectrum, we start to already detect very interesting fragments. Like in this case, for this set of signals here, this corresponds to the Y18 6-. This is a classic sequence fragment, which is labeled according to the McLucky nomenclature. In addition to these familiar type of fragments, we also notice some fragments that do not contain either the 5' or the 3' end of the construct. Like in this case, the mass for this signal correspond to a series of possible trimers, GUC, CGU, UGC, and you notice these could be assigned to different regions of the initial sequence of the precursor ion. If instead we look at a slightly larger type of fragments, like in this case here, this 4-, this sequence here can only be assigned to one specific location in the sequence of the initial precursor ion. And this already shows how important it is to obtain larger fragments that are less fraught with ambiguities when doing the data interpretation. When we first obtained this data, the interpretation was done by using Mongo Oligo Calculator, some freeware available on the web, and a spreadsheet. And we started to plot the possible fragments here against the sequence of the precursor ion. And two things immediately jumped up to us. One was that the fragments, the classic fragment series containing either the 5' or the 3' end of the construct were only few and far between, and they definitely didn't cover the internal part of the sequence. Instead, all these internal fragments that we were able to calculate, once we plotted them here, like these dashed lines, they ended up covering the entire sequence of the construct. If we zoom in some regions here, we also notice that there are many of these internal fragments that cover the same region of the precursor ion. The situation here is very similar to what we can see in next generation sequencing data, where there are multiple reads that cover the same region of the analyte. And this is actually used to increase the confidence in the interpretation of this sequencing type of data. So we believe that the internal fragments can do for tandem mass spectrometry what this depth of reads can do for next generation sequencing data. When we started to do this work, there was no software that will allow us to do this data interpretation. For that reason, I started a company, a private company, to develop new software tools that could capitalize on internal fragments. I developed this app that is called seqRead that can do support for tandem mass spec and also mass mapping experiments. And the app uses an extensive library of non-canonical RNA and DNA nucleotides, so it can really be applied to modify nucleic acids. You should also know that I recently reached a license agreement with Bruker to integrate these algorithms into their software suites. And actually, this is a first example of integration where we can use the data analysis mass viewer to map the different fragments identified by my algorithm to the convoluted spectrum right here. You notice that the majority of the signals here are labeled, except for some of them here that are still unlabeled. And I should say that there is probably still a lot going on upon gas phase activation of these larger ions that we don't understand. So this will be the object of future research. Another way to look at this data is provided again by the data analysis viewer. In this case, in the top panel, we are plotting the different series fragments as well as the internal fragments against the sequence of the precursor ion that allow us to see the coverage. These numbers here represent the discrepancy, the difference in mass between the experimental data and the predicted value from the algorithm. In this view, in the bottom panel here, we plotted all the internal fragments that were detected. And again, you see the extensive overlap of these internal fragments, where there are regions of the initial precursor sequence that is covered by over 10 different fragments. So this type of interpretation, we believe, will provide excellent confidence in the data interpretation here. As I mentioned before, the app works also with non-canonical nucleotides. I'm giving you an example here of the tRNA phenylalanine. This is a 75 nucleotides construct of about 25,000 units molecular weight. Notice here there are several non-canonical nucleotides. And when we perform tandem mass spectrometry, we also obtain data that were very rich in information. We applied the app to perform the interpretation here. And this is the example with the data analysis viewer. And also in this case, we obtain excellent sequence coverage, again, including the modified nucleotides. So, looking at this tandem mass spec of larger nucleotides, now we should ask the question: how do we go after analyzing progressively larger strands? We demonstrated that we can go over 100 nucleotides in size in one shot using what can be called a top-down experiment. But for significantly larger analytes, these would not be possible. And we will have to resort to a typical strategy used also in protein analysis in proteomics, where an initial solution cleavage step will have to be applied before we can perform a mapping and then a sequencing of the ensuing products in there. But this type of approach for nucleic acid can be fraught with challenges. I can give an example here in the sequence analysis of the 18S ribosomal RNA. This RNA has about 1900 nucleotides. If we were to digest this construct in solution by using, for example, RNase T1 that cleaves a G, we would expect up to 541 putative products. Statistically, each of these products will have about 4 nucleotides in average size. So if we think in terms of tetrameric products, like in this case, this ACUG, the same sequence here could be found in at least 5 different locations within the sequence of the 1800 S ribosomal RNA. Similar tetramer with the same base composition but slightly different sequence could be found in many other locations, and we can go on and on in these. All these products will share the same molecular mass, therefore greatly increasing the ambiguity in the data interpretation. So we believe that a possible solution for this mass degeneracy problem will be to limit the efficiency of the cleavage process in solution. So up here you can see, if we were to perform, for example, an overnight digestion with RNase T1, we will obtain presumably a cleavage event at every possible G in the sequence. If instead we were to perform digestion for, let's say, 1 hour, we will start to observe some miscuts. In this case, by skipping some of these possible cleavage sites, we will obtain larger products that would be less prone to ambiguous interpretation. So this can be seen also by using the app to predict possible outcomes. In this case here, if we were to predict the possible products that don't have any miscuts in there, this would be your overnight digestion. You notice that the average size of these products will be perhaps around 10 nucleotides in size. But if we do the digestion where we can have up to 3 miscuts, 5 or even 10 miscuts, immediately we see that the distribution of sizes increases considerably. And we believe that probably the sweet spot will be around here, where the majority of the products that we obtain by cleavage in solution will have a size below 25 nucleotides. This size is very readily amenable for tandem mass spectrometry and will really provide the best value for this type of experiment. So this is an example of an experimental example of analysis. In this case, we perform digestion of the 18S ribosomal RNA for less than 1 hour. The spectrum that we obtain is particularly rich of information right here. In this case, we're still working on the integration of the algorithm with the data viewer from Bruker, but we can already obtain results here where these are the matches between the experimental data and the predicted fragments. You see that our app can also predict not only regular RNA products, but also their possible salt adducts produced in solution. And with these, now we can already start to label the spectrum here and look at the coverage. So at the end, what I would like to state is that by using this strategy, we can perform what has been called for protein analysis a mid-down strategy, where we limit the solution cleavage step to obtain larger fragments that are less prone to ambiguous determination. So at the end, these should be my take-home points for the data that I showed you today. We demonstrated that it's possible to perform top-down analysis for precursor ions that are greater than 100 nucleotides. The spectra obtained from these larger precursors is dominated by internal fragments, and we had to design new software to be able to interpret this type of data. And the software that we design, as I said, work for both normal and non-canonical nucleotides. The software can also support mid-down approaches, both in the planning phases and in the interpretation phase. And everything that I just showed you was carried out by using direct infusion analysis using static nanospray, but the tools and the concepts are readily adaptable to a CMS application. So we believe that there is a lot of value in these type of strategies. And at the end, I would like to give special thanks to the people who carried out the work, Tom Kenderdine and Will McIntyre at UConn, Chris Bell at Ribodynamics, Guillaume Trementin, Heiko Neuegger, Alexander Bankowski, and Michael Craig at Bruker. These are my sources of support, and primarily NIH, and special thanks to the Harold H. Schwenk Senior Endowment. And if you need to contact me for experimental issues about the digestion procedure and so on, you can contact me on my UConn email. If instead you would like to talk about software issues, you should use my Gmail account. And with this, I would like to thank everybody for your attention, and I will be happy to answer any question that you might have.